Thank you very much. Thanks to New Phytologist for the invitation. I'll be talking about roots. So it was great this week to hear that there seems to be a consensus that roots are important. So I don't need to convince you that they are important. So that's great. But on the other hand, if that pointer worked, yes. If you look at the data that's available, and you all know that, if you look at the fine root data, the number of observation in dry, well, there are actually very little compared to the leaf data, reproductive traits, etc. So it's a little bit better now thanks to the efforts of Luc with Fred and Catherine and other people in this room, but still traits are underrepresented. And really, I don't know what the fuss is all about, because once you find PhD students, it's actually quite easy to go out in the field and measure root traits. But no, on a more serious note, it's very difficult work, and I've seen a lot of enthusiasm for measuring root traits, which is great. And but I've also heard that there's some confusion. Ian mentioned that roots confuse him, so I hope that I won't confuse you further. But to do this, I'll try to provide maybe some kind of suggest a possible roadmap and maybe avenues for further research if we want to advanced below-ground trait-based plant ecology. Now, I made some suggestions in a recent paper. I don't want to talk about all of it because other people in the room will do a much better job at this, and also I don't have time. But I will focus on 3 things today. So about trait environmental linkages, so traits related to nutrient use, nutrient acquisition. So I'll talk a little bit about leaves, but mostly about roots. I will also talk about what I think is the elephant in the room. So mycorrhizals, everyone knows they're there. They're important. They play a major role in nutrient acquisition, yet they're often ignored. So I think that's the biggest challenge. I'll be talking about some of those challenges. And then I'll be talking a little bit about how below-ground traits can help us understand emergent properties of ecosystem, but I'll be focusing mostly at the community scale and on plant species diversity. So to do that, I want to illustrate that with some of our own field research. That field research was quite local, so compared to all the global analysis. So this is a very local scale study, and it was done, well, all the studies were done in this part of the world, southwestern Australia. And there are 2 main reasons why I think it's a more interesting and useful study system. The first one is that it's a global biodiversity hotspot for plants. So if you look at where most of the plant species are in the world, where they're mostly around the equator, as you know, but there's a little outlier here in southwestern Australia where the number of plant species is very high. Now, the second reason why I think it's an interesting study system is if you start looking at soils, there's a wide range of geology, but also soil age. So all along the coast of southwestern Australia, so from it's about a 400-kilometer distance north and south of Perth, you have the Swan Coastal Plain, and you have dunes of different ages that range from present day, so all across the Holocene. But as you go inland, you find dunes that are up to 2 million years. And so why is this interesting? Well, it's interesting because it controls for, well, minimizes variation in geology, parent material, certainly minimizes variation in climate, and it creates a wide range of soil nutrient availability. So it's an interesting natural experiment to look at the effects of soil fertility on plant communities and their traits. So what do they look like? Well, if you go in the really young dunes, so it's really primary succession. So what you see, plants coming in, you have new parent material being deposited on the dunes. So what do you find in terms of nutrients? Well, as you would expect from models of pedogenesis, so if you're in that, and you'll see a lot of these graphs with chronosequence stage, but you can interpret it as just age of the soil, so 1 being young and 5 or 6 being old. So when you start in really young systems, there's very little nitrogen, because perhaps as you know, nitrogen initially is not present in most rocks. There's very few rocks that contain nitrogen. It comes from the atmosphere, so it needs to be fixed and incorporated in plants and soil. So initially, there's very low nitrogen. And in terms of the phosphorus, well, phosphorus is a rock-derived nutrient, so you've got all the phosphorus you need. So initially the system, well, plants are limited more by nitrogen availability. Now if you give a few hundred or a few thousand years, then what you end up with is buildup of nitrogen. So nitrogen builds up relatively quickly within centuries or decades, and then you're at the highest that you'll ever get. Phosphorus is still relatively high because it hasn't had time to be lost. So you're probably at the peak fertility, and it's at that stage where both nitrogen and phosphorus kind of co-limit plant growth. So if you give a long time for pedogenesis to move forward, what you end up with is you gradually lose phosphorus in the system. And eventually, in that case, after millions of years, you end up with very little phosphorus. In fact, once we measure the phosphorus concentrations in the soil, this is total phosphorus, it's the lowest that's ever been characterized in the world so far. There are a few other places that sort of match it, but I think it's fair to say that these are probably the most infertile soils on Earth. So we've done some experiments and we've shown pretty clearly that their phosphorus is the limiting resource and plants struggle to acquire that limiting resource, and it's really what drives productivity and perhaps competitive interactions and so on. So I want to come back to this and illustrate trait-environment linkages, mostly across that gradient. Perhaps we can learn something for global models and etc. So what happens to leaves? Well, that's the first thing we did because that's very easy to do, as you probably know. So if we look at, for example, leaf phosphorus concentration, and that's an average, a community-weighted average for different plant species. So initially in the very young soils, the leaf phosphorus concentration are about what you'd find in a crop plant, so quite high. Plants are happy. And as pedogenesis goes on, eventually in the phosphorus poor, really old soils, we end up with leaf phosphorus concentrations that are the lowest recorded on Earth, really. So it's a very nice and clear pattern of declining phosphorus concentrations across that gradient. Now, when we look at senesced leaves still attached to the plant, we find that the same decline, but the proportion, the difference between the green and the senesced increases, which means that the plants are very efficient at resorbing, recycling internally phosphorus, and they recycle about 80 to sometimes 90% of it. The leaves are also long-lived. They live several years. That's an example of a Banksia species. And if you look at the leaf anatomy, they're very well defended. So these are segments of mesophyll surrounded by lots of fibers and other things to make the leaf very tough and long-lived. So if I come back to Sandra's slide from yesterday, she presented an image of Hekia. Well, that's a Banksia, but it's fair to say that it would fall, at the margin of this galactic plane, certainly in terms of the leaf economic spectrum. So it's so she mentioned that it was perhaps interesting to look at what happens in the margin, where we have an example where we can do that. But what about the roots? Well, of course, we know that roots are not represented on that galactic plane, but we can have a look at what happens. Sorry. Whoop. There you go. So when we look at the roots, well, we actually find the roots are very short-lived, and they're very poorly defended. And so I would argue that perhaps they have tough leaves, but they have weak roots. So if we look at root anatomy, there are no obvious structural defenses. A lot of studies have been done on root development. So they make these cluster roots, so not all species, but the Proteaceae do. And these roots only live for a few weeks. They have a peak around the middle here after a few days. What they do is they essentially mine phosphorus in the soil. So they exude a massive amount of carboxylates, and this mobilizes the phosphorus in the soil solution, and then they can take it up, and then there's no more phosphorus, so they move on. So, and they're non-mycorrhizal as well. So maybe that's sort of counterintuitive. We have very well-defended leaves, poorly defended roots. So what happens? At first, maybe it looks counterintuitive, but really when you think about it, above ground and below ground resources are very different. So on one hand, if you look at leaves, they get light, they get CO2. So these are resources that a leaf cannot locally deplete for itself. Sure, it can take some of the resources and shade other leaves around, but as it takes photons, the sun keeps sending photons, and as diffusion of CO2 in the air, you can take CO2, but more will come to your leaves. So there, maybe if you want to conserve nutrient, it makes sense to make a leaf that will last many years. But roots, on the other hand, well, it's a resource that gets locally depleted very fast. And we all know that from a lot of agricultural studies. And that's especially true for phosphorus because phosphorus moves very slowly in soils. And so it gets depleted around the roots very quickly. And so, to me, it doesn't make much sense to have a long-lived root if there's no more resources to acquire, right? So, in one way, maybe you could say that the root is acquisitive, right? Because it's very efficient. That strategy of exuding carboxylate is very efficient. So maybe it's acquisitive because it's very efficient, it's short-lived, it's not protected. On the other hand, there's one thing we haven't really looked at is how nutrients are recycled in fine roots. Because, the long tissue lifespan is one way to increase the mean residence time of nutrients, but resorption is another one. That's hardly been measured, well, not very often in leaves, but certainly not very often in roots. But there are very few data, so that's data from some of these non-mycorrhizal species, and as you can see, within a few days, the phosphorus concentration declines to extremely low level. So they're still able to resorb most of the phosphorus before this senesce. So perhaps in one way they're conservative, even though they don't live long. Something to think about. All right, I want to talk a little bit about the elephant in the room, mycorrhizal, and I'll talk about a few of the challenges. So one of the challenges is that it's a trait that can be quite plastic. So for example, this is an extreme example, but we have plants that form dual associations, sometimes on the same roots, with ectomycorrhizal fungi and AM fungi, arbuscular mycorrhizal fungi. And we've done studies growing them in the different soils, the same species, to see whether there's a switch. And for the two species we studied anyway, initially in the young soils, they start mostly with AM colonization, and the theme is about trade-off. There seems to be, anyway, a switch from as soils age and phosphorus declines to colonization more by ectomycorrhizal fungi. So quite plastic and certainly a challenge for below-ground trait studies and integrating mycorrhizals. Another thing is that plants don't associate with only one species. Generally, specificity is quite low. And so if we've done studies looking at the mycorrhizal fungal communities, so which species of fungi you find on the roots, and even on the same plant species, you can find completely different communities. So for example, this is ectomycorrhizal fungal communities on the same plants, and very high turnover in terms of the fungi. So we don't know what the traits of the fungi are, but they might do something very different in terms of function. So we've looked at it at the community scale more as a categorical trait to see what's the relative abundance of these non-mycorrhizal plants that I mentioned were very efficient at getting the phosphorus, and mycorrhizal plants such as arbuscular or ectomycorrhizal. So in the young soils, you start with quite a low abundance of these non-mycorrhizal plants, but as pedogenesis goes on, they become more abundant, and mycorrhizal abundance goes down. So I told you they were more efficient, but when you look at it, there's about an equal proportion of these non-mycorrhizal and mycorrhizal. So certainly all the strategies are still there. Oops, sorry. So I want to come back now to go at the community level and explore whether there might be potential trade-offs between different nutrient acquisition strategy. And one of the things we thought about was that there might be a trade-off between being very efficient at acquiring the phosphorus, for example, those non-mycorrhizal species that have these short-lived roots, but perhaps the trade-off is that they don't have high defense, they're poorly defended against pathogens, and that might have interesting ecological implications. So one of my students ran some Glasshouse experiments, and what he did is we first isolated some native pathogens from these soils. So we think that oomycetes, in particular species of Phytophthora, are quite present in these sandy soils and might be an important pathogen that doesn't necessarily kill the plants but acts more like a herbivore. So first of all, we grew these non-mycorrhizal cluster-rooted species in competition with ectomycorrhizal species in the absence of pathogens. And there, as we expected, the non-mycorrhizal species grew better, and it sort of outcompeted in a way the ectomycorrhizal species. It certainly was able to take more of the resources, and the ectomycorrhizal plant was suppressed. Now, when we did the same, but we added those pathogens, it didn't kill the plants, but it shifted the competitive balance. And then suddenly, the non-mycorrhizal species were not doing as well, and the ectomycorrhizal species were doing as well. So it's kind of an example of how these trade-offs can play in terms of competitive interactions. Now, we also wanted to look at it from a longer-term perspective and more involving multiple generation of plants through something called plant-soil feedback. So we proposed in 2015 a hypothesis how these nutrient acquisition strategies could influence plant-soil feedback. So on one hand, maybe this is one extreme, a non-mycorrhizal plant. If it's poorly defended against pathogens, what might happen is as the plant develops, it's a resource for pathogens, so you end up having a buildup of pathogens in the rhizosphere. Now, most seeds would fall quite close to the plant, but the seeds of the same plant fall and germinate in an environment that's not a very good environment for recruitment because it's exposed to these pathogens that perhaps are a little more specific to that species. So in that case, what you'd find is that recruitment would probably be better away from the mother plant than close to it. And this sort of negative feedback can help promote coexistence because it prevents a species from becoming monodominant. Now, at the other extreme, perhaps we have something different, and that's been observed in even species-rich tropical forests where sometimes you have these monodominant stands, and they're often ectomycorrhizal species, not always, but often they are. So perhaps there's something going on with ectomycorrhizal species. And we find something similar in southwestern Australia. Sometimes we find patches of ectomycorrhizal shrubs that seem to form, and we don't know why, because we look at the soil properties, they're very similar. So we think something that might be going on is the mother plant invests a lot of carbon below ground into a mycorrhizal network, and then it offers quite a nice environment for a seedling to germinate because it can associate straight away with the right symbionts, and perhaps offer some protections pathogens because it's well known that mycorrhizal fungi, especially ectomycorrhizal fungi, offer protection against soil-borne pathogens. So in this case, this might be a positive feedback. So we wanted to test some of these ideas, and we did so with a Glasshouse experiment. So it's quite a large experiment. We went in the very phosphorus-impoverished soils, species-rich areas we could find, and then we selected a bunch of species from 5 different nutrient acquisition strategies. So we had different types of mycorrhizal plants, non-mycorrhizal plant, nitrogen fixing, and so on. So what we did is we grew the plant in the same field soils that we first sterilized, but then we re-inoculated with a tiny bit of soil, and the soil either came with sterile soil, or it came from its own species, or it came from plants from the same strategy but not the same species, or plants from another nutrient acquisition strategy. So it became a large experiment, and thankfully I had a hard-working postdoc and lots of students to run an experiment with 1,000 pots, which I'll never do again. So what we end up finding is, it was sort of consistent with the model we proposed. Of course, it's never as clear when you look at the data, but still, when we look at the non-mycorrhizal species on average, there seems to be evidence for negative feedback. So survival when growing in its own soil was often poorer than when growing in other soils. That was also true for nitrogen-fixing species, which was interesting. On the other hand, when we looked at ectomycorrhizal species, we found evidence for positive feedback. So meaning that either survival and or growth was often better in its own soil. For our vascular mycorrhizal species, we didn't find any significant feedback, but we only had a few species, so perhaps low sample size. But what I think was more interesting is then we used the growth parameters and the survival parameters from the Glasshouse data. And we ran some simulations to see what are the potential implications for local plant species diversity. So we had different scenarios, one of which was we grew the plants as if there was no soil biota in the ground. So what happens then is that if you look at Simpson diversity, then it declines very rapidly to unrealistically low levels. You never find these low levels in the field, they are rarely. So in scenarios that integrated both the effect of a plant on itself, as well as the effect of other strategies and neighbors. And what we found was that it actually contributed to promote diversity for longer. Of course, it's just a simulation, but at least it shows that perhaps the potential is there for these plant-soil interactions to maybe play a role in plant species coexistence in these environments. So I just want to come back and maybe have a short summary and maybe offer some perspectives on these three things, or there are only two there, that I talked about today. So first, trait environmental linkages and mycorrhizals. I'll talk about it together. I think this is very important because as we've seen, we have very few studies, very little data on how root traits change across nutrient gradients. There are very few studies that have controlled, well-characterized nutrient gradients. So I told you mycorrhizals is a big challenge, and there's a lot to do, but perhaps as a first step, root colonization is something useful that we can relatively easily measure. It doesn't tell you everything, but I think it's a good first step. And as Luke mentioned, get more information on root anatomy and root morphology for fine roots. And as it happens, I have a student doing that, which is great. And very preliminary results, but on another coronal sequence that we have further south, what he found in the same species, so we talked about intraspecific variation. So in some of the more fertile sites, he finds quite high arbuscular mycorrhizal colonization, and when he goes in the phosphorus poor sites, well, the roots have no mycorrhizals whatsoever. So it shows that it's actually quite plastic. I think a next step, and I don't think I'll be doing that myself, but I hope others will and colleagues, is perhaps to go one step more and measure the hyphal traits. So because it's the hyphae that do a lot of the nutrient acquisition in many situations for many plant species. Now that will be very difficult, but it's not impossible. Some people have already proposed frameworks to do that. One of my colleagues, Pierre-Luc Chagnon, proposed that we could perhaps apply Grimes' CSR framework to arbuscular mycorrhizal fungi. That's a while ago, but people have talked about using ectomycorrhizal exploration type as a categorical trait, perhaps something that tells us something about where the distance, how fungi acquire nutrients from soil. So that's something that we might have to think about. Lastly, about using traits to understand emergent properties of communities, I think there's a lot of further work that can be done there. We often think about root trait diversity in terms of resource partitioning, and I think one of the perhaps interesting thing in our studies, we showed that there's another side of the story is that nutrient acquisition strategy can determine the response to pathogens, and that might play a role in promoting plant diversity. So it's another aspect that I think hasn't received much attention so far. So with that, I just want to thank New Phytologist Trust for inviting me, and funding agency, and a lot of people, students, postdocs, and colleagues that have helped me with this research. Thank you.